Not bad. Yeah, so preferred administration for pharmaceuticals is through the oral route. So that's going by your mind-me kind of like creed, I guess. So it's oral, like method of administration should always be through the oral route. It just makes it easier. You can tell by like how many drugs are through the oral route when you go to the pharmacy. No one's going to give you an IV. And topical is more for superficial effects. You can't really get too many systemic effects, and you can't affect a lot of things through the topical route. So oral can affect most things. Good. So osmosis. Osmosis is just the movement of water. Yeah, movement of water, and drugs aren't water, so therefore drugs can't move via osmosis. Yeah. Yeah, sweet. Okay, yeah. Cool. So first intention is just like a paper cut, and then it heals pretty well. Nothing too drastic. Yep. So chronic stress. So what is a reason? What is not a reason why chronic stress is stressful? So demand refers to things like it's demanding a lot of your time or like your brain power to do. Controllability is, well, chronic stress is generally not that controllable, I think. So you can't really help it. So it's going to contribute to the chronic nature of it. Predictability, probably not that predictable as well. That's why it keeps on going. Preventability. Wait, what does this refer to?
It's not a thing.
Yeah.
I made it up.
Yeah, preventability is just not a thing. Yeah. Yeah, I think these are like the dimensions of stress or something like that. So yeah. Good. So yeah, primary appraisal is like the first step where you decide whether it's actually stressful or not. Is it relevant to myself? Is it positive? That kind of thing. Stress is like, it's the outcome where you evaluate your resources versus the actual stressor. And then depending on the balance between those two things, you can get stress or you may not get stress. And coping is what you balance your stress against. So do you have enough coping strategies to deal with the stress, or are you kind of stuck and you don't know what to do? Yeah, so all of the blue, red, and yellow are parts of the coping model. So it is none of the above. Sweet. Yeah, so all of these Yeah, so all of these are maladaptive. So maladaptive meaning they don't help. So denial, you're kind of just avoiding the problem. I think that's the one that's probably the trickiest, I guess. But yeah. Yeah, nice. So external attribution, you want to think external to what? So it's external to people. So therefore it would be your situation around you. I think this is one of the questions I shouldn't have explained. Oh no, that's fine. Disposition refers to like your own disposition to do something. So like it's a bias of yours. Yeah, nice. So this question was kind of like a statement. It's like a viewpoint that someone might have, and you have to think about, yeah, what type of attribution is this? So the key in this one is entire life. So that's like affecting every single part of it. So therefore it's global. That's kind of the opposite of specific, because it's not like it only affects their ability to, I don't know, play a sport. It'll affect everything. Stable. Stable, I guess the statement didn't really touch upon. And then controllable. Being paraplegic isn't really controllable, unfortunately. So a similar style of question here as well. Yep, sweet. Yeah, so this one here is like, so we're kind of thinking about the mood and how it's all pointless. So if something's pointless, if like some action they're doing is pointless, then that means that they can't take control of it. It's like taking the tablets isn't able to control the mood. Yeah, so unstable, I believe unstable kind of refers to the frequency and like the timing of things. So this might be like, sometimes my mood affects something, at other times it doesn't. Those are kind of like unstable things. So talking about how often they come up, rather than whether it's controllable or not. So the mood, like the tablets not being able to help the mood, makes it uncontrollable. Yeah, because they're trying to do something, but they really can't do anything. Does that answer your question? Like, does that make sense why it wouldn't be unstable? All right, sweet. Yeah, nice. Fundamental attribution error. Oh man, I don't remember this. Hey, David.
So essentially fundamental attribution error is, so actor observer bias is like when you're the actual person acting and you think like, oh, I'm so stressed at work because my boss is
Don't say the actor one because that's the next question.
We'll explain it after the next question. We'll explain after the next question, David. Yep.
There we go.
Okay, go for it, David.
So active observer bias is more like you're the actor, and then you say, "Oh my God, like I'm so stressed because of external factors." Whereas fundamental attribution bias is that, let's say you see someone homeless on the street, and you immediately say that person must be like a drug addict or have a lot of things, and you immediately put on like a certain narrative for them to explain their current situation. Or in a more medical sense, you see a patient with depression, and you can say, "Oh, it's all just in their head," and a lot of it comes from like kind of cultural influences on why you see things like that, and then it's like important not to, I guess, essentially jump to these conclusions.
Yeah. Yeah, so self-serving bias is like trying to make yourself seem better. So self-enhancing is like having a super inflated ego. So like you're trying to really push yourself on, I mean, saying, "Oh, I'm so great, I can do all of this." So that helps you like think better of yourself. Self-handicapping is like, I think this one is the one that David was saying, like a lot of med students do. So it's like, "Oh, I haven't I've hardly studied for this. I have no idea what like any of these statistics mean. I have no idea how to make this graph. Like it doesn't make sense. I'm going to fail this exam." And then you get your exam results back, and then they're perfectly fine. So you've handicapped yourself, even though there's no need to. And then self-protecting is like, "Nah, I don't think I did that well on the exam," and you try to lower your own expectations, so you're not as cut afterwards. And then self-justifying, I don't think exists. Yeah, that just, yeah, it's false. Nice. I won't explain this yet because, oh, what? Oh, well, I think most people got it. Okay. Yeah, so viral antigens, viruses are like intracellular. So I like to think of them as being inside the cell. Inside, like the eye looks like a 1. So MHC class 1. Class 2 is like for your more extracellular kind of things. If a immune cell like phagocytoses is a bacteria, those antigens will be shown on MHC class 2. Whereas if a cell is infected with the virus, that'll be on class 1.
Yeah, so it also, because class 1 is like, the rule of 8. So when you activate class 1, you activate CD8, and CD8 is used for intracellular. Yeah.
Yep, that's your rule of 8. The other one being your MHC class 2, activating your CD4, which is for extracellular, like through your antibodies.
Mm-hmm.
Nice. So antibodies, you want to think of antibodies as like helping your immune response, or trying to help the immune response, or trying to prevent abnormal growths. So helping the immune response, promoting phagocytosis can do that because you're like eating up all the bacteria, right? Cell activation, I think refers to you're like kind of alerting cells to the virus, I believe. And then activate complement proteins. So complement proteins are those soluble proteins that kind of circulate, and they have really a variety of functions, but mostly to strengthen the immune system. It wouldn't make sense for antibodies to promote cell proliferation, mostly because if you think about the immune responses response to like a tumor, you don't want your antibodies to encourage that tumor. So you don't want cell proliferation. So logically, having an antibody triggering cell proliferation wouldn't really make sense. So it's not a function. And then last question, which I think we might have explained, but yeah. Oh, no, we haven't. It's okay. Sweet. So Th1 is for intracellular. Th2 is for extracellular. I think this one, once again, I believe you can use your rule of 8. So your Th1, 1 times 8, or your CD8, is for intracellular, and then your Th2 is for more extracellular. So 2 times 4, and your CD4s are for extracellular. So Th1 cells are for intracellular pathogens. Helminths are like a type of extracellular pathogen, and so are nematodes. So even if like you see this in an exam and you're not too sure, if you can figure out what a helminth is and what an extracellular pathogen is, if both of them are correct, like if both yellow and blue are the same answer essentially, they can't be the answer because you can't have 2 answers in a single multiple choice question, if that makes sense. But yeah, that's the end of the quiz. Does anyone have any questions about any of the questions in the quiz? Otherwise, we might let RJ present because Mans is sleepy. Whoa. All right, sweet. Good job, smiley face. So the rule of 8, when was that? That was like quite a while ago. But the rule of 8 is to help you combine which MHC class goes which, oh, I cannot speak. It's to help you pair up the MHC class and the
T cell.
Yeah. So you have 2 MHCs, right? You can either have MHC class 1 or MHC class 2. And then you can have 2 types of T cells, CD4 or CD8. And the rule of 8 is that if you consider the 2 numbers in a pair of like MHC and T cell, the product of those 2 numbers must equal to 8. So that means that you have to pair up your MHC class 1 with your CD8 because 1 times 8 is equal to 8. And therefore you have to pair up your MHC class 2 with your CD4 because 2 times 4 is 8. So simply just to help us to remember which MHC goes with which T cell. And that's the rule of 8. Cool. Any other questions about anything in the quiz? Otherwise, I think you can get started, RJ.
Yeah.
Okay, antibiotics. Have you guys had your antibiotics lecture yet? Like your whole antibiotics, antivirals, antifungals? Yep, okay. Rough or pretty okay?
Yeah, honestly, it's a lot to learn for antibiotics, and they keep it till the end of semester. So, and especially since it's your first time learning about drugs and pharmacology, it might appear intimidating, but over time, you'll have, I guess, you'll figure out your own way of actually like learning different drugs, different drug names, their mechanisms, and um, for your very first time learning pharmacology, I've tried to like put some memory tools so it's easier for you. But over time, you'll actually start learning it by its mechanisms rather than using memory tools. Okay, so antibiotics in therapy. So there are a few key terms that you need to know. Firstly, if you use in therapy, you've got chemotherapy. So that's the use of chemicals to treat and control diseases. And you also have prophylactics. Prophylactics are to prevent infection when there's a significant risk, especially if you're immunocompromised. And antibiotics, there are two classes. So there's an empirical and directed. So empirical is a broad spectrum kind of drug. So in this case, the diagnosis is not certain, so you're not sure of what the organism is. So you use a broad spectrum of antibiotics to target different strains of bacteria or any type of organism. And a directed is when it's a narrow spectrum, where you've actually taken a culture of the pathogen, and you're actually able to determine what's the pathogen, and so you use a more specific antibiotic. So each of these antibiotics have its own benefits and disadvantages. But basically, if you know the pathogen, you use directed. If you don't, you use a broad spectrum antibiotic. So there's a few pharmacokinetics of antibiotics. So you've learned about pharmacokinetics, which is the effect of the body on the drug. So the dose depends on the therapeutic window index, which just refers to the range at which there's a minimum effective concentration and minimum toxic concentration. So that's therapeutic window. And your volume of distribution. So your volume of distribution just refers to the volume required to make the drug present in the body as the same concentration as in the plasma. So imagine if you were to take a drug, an antibiotic intravenously, right? And if this drug was to continuously diffuse out of your bloodstream into tissues, basically if it doesn't stay in your vascular system, you'll require a really high volume in order for it to reach the same concentration as that of your plasma. Whereas if it doesn't leave your bloodstream as quickly, then you don't need that much volume to reach your plasma concentration. And that's basically the summary of this formula that you need to know. So if it has a low volume of distribution, it will stay in your vascular system, it will stay in your bloodstream, it won't diffuse out. But if it has high, it just means it keeps leaving your bloodstream, and so you need to keep on replenishing it.
Right. So just clarifying the whole empirical and directed drugs thing. I think someone said the question. So
Oh, I didn't see it.
empirical
Sorry.
is when the causative organism is not proven. So let's just say you feel sick, you've obviously got some infection, but we don't know if it's like Staph aureus or if it's, I don't know, Strep viridian. So we don't actually know exactly what the organism is. So that's why we give a broad spectrum, and that's called like empirical therapy. So what we might actually do is that we might originally give someone empirical therapy where the organism is not proven, so we don't know which microorganism actually is, and then like get a sputum culture or something like that, and then figure out what the actual organism is, and then switch to directed therapy, which is more narrow spectrum. Hopefully that makes sense. So it's not to do with the mechanism, but it's more to do with the actual organism.
Yeah. And sometimes you like what Zoe is saying, you give an empirical first, then a directed. But there are times when you rather wait out for the culture before you give the antibiotic. So then you just give a narrow spectrum in that case. So moving on to frequency of antibiotics. So your frequency of dosage of antibiotics actually depends on quite a few factors. So firstly, there's half-life. So that looks at your drug and like the half-life, so which is like the amount of time it takes for it to be reduced to half the concentration. Then the time to maximum concentration, so that depends on the method of consumption. For example, like if you were to ingest it, it'll take a much longer time for it to pass through your digestive system and into your bloodstream. Whereas if you were to take it intravenously, it'll hit maximum concentration much faster. And also, it also depends on your toxic levels. So if it has a low therapeutic window, it means if it hits your max concentration, anything higher than that, it'll be too toxic for your body. And then there's other factors such as elimination time, the time taken for your body to remove the drug, the post-antibiotic effect, which just refers to how like sometimes once you take a drug, it can actually cause continued suppression of bacterial growth within the body. And this is the case for certain mechanism of drugs, certain antibiotic types. So there are certain antibiotics which target like cellular replication within pathogens. So if they target the replication, it actually suppresses bacterial growth because like new bacteria can multiply, can grow after once you give the original, like the first dosage. So that's the whole idea of post-antibiotic effect, where even after the concentration of drug in your plasma has dropped, it still has an effect on your pathogen. And there's also a few other adverse effects. So if you were to be allergic to the drug, or if it causes other symptomatic issues such as vomiting, nausea, you also wouldn't be taking too much of these drugs. And there's also other drug interactions. So certain antibiotics react with other antibiotics, or they may react with other types of classes of drugs. So that's the pharmacokinetics, and there's also pharmacodynamics of antibiotics, which is the effect of the drug on the body. So there's two types that you need to know. There's bacteriostatic and bactericidal. So bacteriostatic prevents growth, and it's only useful when the immune system is active. And it basically targets the cellular replication pathway. So it can target your DNA or protein synthesis. And so that prevents the growth. But bactericidal actually kills the bacteria because it targets your cell wall instead. And the important thing that you need to know is that sometimes when you kill the bacteria instead, dying bacteria can release endotoxins, and that can be potentially more harmful to cells. So that's why sometimes you may not give bactericidal for a particular type of pathogen or bacteria. You rather give bacteriostatic. Okay, so you'll be learning about 4 kinds of mechanisms for antibiotics, and from there there'll be different drug types. So people like you all may have different styles of learning for these drugs. There's quite a few drugs that you'll be learning within these 2 weeks, and it's kind of confusing. So I've grouped it under the 4 different mechanisms of actions, but this is not it's not the only way of learning it. Some people don't group it under this way. They may learn it by rote memory. They may learn it by other memory tools, but I've just tried to group it within mechanism of action because it's how I've learned it, and it makes kind of quite a bit of sense to me, and hopefully it makes sense to you as well. So the four ways is firstly protein synthesis. So it disrupts protein synthesis. It can also disrupt folate metabolism, cell wall synthesis, and nucleic acid synthesis. So you don't really need to know in detail how much it actually affects all these different pathways, but you just need to know there's four mechanisms. Within that, there's drugs, and it helps you understand which drug you use for a certain situation. Okay, so moving on to just specifically cell wall synthesis disruption. You've got your transpeptidases. So that's known as your beta-lactams. So you've probably heard of penicillins, and there's also this other drug class called cephalosporins. Then you've got transglycosidases, which is glycopeptides, and you've also you also use certain bacteria to affect the mycolic acid in Mycobacterium. So that mycolic acid in Mycobacterium just refers to tuberculosis. So tuberculosis is a very special disease where you use a combination of different drugs. And so one drug actually targets the cell wall of the TB bacteria. So like all these just seem like lots of foreign names to you. So I've tried to break it down even further. So focusing just on transpeptidases. So transpeptidases are beta-lactams. So beta-lactams, looking at just penicillin alone. So I've tried to make a table for each drug, and hopefully that helps you understand each better. And I've kind of made a memory tool. Like these are very silly ways of memorizing things. And but especially since it's your first time learning it, I think the sillier the memory tool, the easier for you to remember it. Yeah, so firstly, let's start with penicillin. So penicillin, the specific drug that you need to know under the penicillin drug class is called amoxicillin. So the mechanism of action is that it irreversibly inhibits transpeptidases. So you just need to know it's irreversible. And there's a few important, basically this is like a high yield slide. I've gotten most of the main points. You'll be probably learning more about it. So firstly, it's first line, and it's effective for gram-positive bacteria. And times you often you often get this drug called augmentin from doctors when you're prescribed penicillin, and that's usually when you have amoxicillin, which is the drug, combined with clavulanic acid. Okay, and there's a few side effects of this drug. So certain patient populations may be hypersensitive, and so they are allergic to this drug, and the allergy could range from maybe a rash to even something severe as anaphylaxis. And on top of that, because this drug is quite strong, it can also cause super infection. So in certain areas such as it can cause oral thrush, vaginal thrush, because it can kill the bacteria present in those areas and cause a super infection. And so just a memory tool. So amoxicillin, penicillin, right? You just you can remember it as being the most used. So it's like the most powerful. So like, what would you associate as the most powerful? Like probably like Superman, right? So Superman is like the most powerful being, and so you'll have hypersensitivity and super infection. So just associate those two things with amoxicillin. Okay, so the next drug under beta-lactams is cephalosporin. So that's a drug class, and then the specific drug that you need to know is cephalexin. And again, its method of action is irreversible, and usually you give this drug when you have got patients who are somewhat penicillin resistant. So if the penicillin resistance, like if they are really allergic to penicillin, you wouldn't be giving this because they're under the same group. But if they are slightly resistant to it, you can possibly consider this. Okay, so again, so memory tool, so cephalosporin, so spore is like part of a mushroom, right? So usually mushrooms can be toxic and cause infections. So that's why this drug can lead to opportunistic infections, or even hypersensitivity. And one main thing that you need to know is that it causes neurotoxicity. Okay, so moving on to your next mechanism, within your next mechanism of action, which is your transglycosidases. So under that, you have your glycopeptides, and vancomycin is a specific drug that you need to know. So again, that's irreversible. And the main thing about vancomycin is that you use it for MRSA and MRSE. Basically, use this drug for bacteria which are usually resistant to other types of antibiotics. And sorry, I'm supposed to take out the last point. It's not supposed to be there. Yeah, remove the part about the not if severe, because that's the previous slide. Okay, so adverse effects. It causes red man syndrome. Basically, your face turns red, and it can lead to nephrotoxicity and ototoxicity. And you take this intravenously. So a memory tool is that just remember a red man called Mr. SA driving van. Okay, so red man is red man syndrome, driving MRSA, and use vancomycin. Like, some of these memory tools may not be the most ideal, like for you. Well, you can think of your own one. I'm just trying my best to come up with some silly ones. So so like I was saying about TB. So moving on to TB, there's a few different drugs that are used for TB and use them in combination. And so one specific drug that targets the cell wall is called isoniazid. And that, sorry, it targets the cell wall. And the cell wall of TB is different from most bacteria because it contains mycolic acid instead. So this drug actually inhibits the synthesis of that acid. And so it's the first line therapy of Mycobacterium, which is the bacteria for TB. And it's used in combination with other TB medications. And you'll be learning about RIPE, which is the acronym for the different medications that you use. Okay, so the adverse effects, hepatotoxicity and hepatitis. So this is really silly. So the memory tool is isoniazid. So you see the word son in it. So that's he. So that's like him and he, right? So that's hepatotoxicity and hepatitis. Yeah. So moving on. So that's all you need to know for cell wall synthesis and the different bacteria you need for that. And moving on to protein synthesis disruption, you've got quite a few drugs for that. So you have got certain drugs that target the movement of the ribosome. You've got certain drugs that target the reading of mRNA, the binding of tRNA to mRNA, and the transfer of the peptide from the tRNA. So again, I'm just going to give you tables of these different drugs. So macrolides, that's a class of drugs. And the specific drug you need to know is erythromycin. So just need to know it reversibly binds to the 50S subunit of the ribosome. And usually you use this for gram-positive bacteria, especially if it's penicillin sensitive, and for upper respiratory tract infections. Okay, so for this, it's macro slide. So imagine like a 50-story slide. So when you're sliding down that slide, you'd be probably really scared. So your heart skips a beat. This drug actually causes arrhythmias, and you'll probably definitely feel a nausea. So you'll have GI upset from this drug. Yeah. That's it. I think that's the main things you need to know: arrhythmias and GI upset. Okay, so next one is aminoglycosidases. So the main drug you need to know is gentamicin. So this one is just, you have to remember it's irreversible, and it has a broad spectrum, and it's targeted for gram-negative aerobic bacteria. Okay, so the adverse effects is nephrotoxicity and ototoxicity. So the way I remembered it is, gentamicin is like gentleman. So you can try to picture like, the character from Up, the gentleman from Up, and what would you associate him to be? He'll be faithful to his wife, so that means it's irreversible, and um, um, like when you're old, you probably have hearing and kidney problems, and on top of that, usually when you're elderly, you might have breathing issues, so you'll be on a ventilator. So when you're on a ventilator, it just shows that you need the need for oxygen, right? So that's referring to bacteria which requires oxygen. So it's targeted for bacteria which requires oxygen to grow. So it's gram-negative aerobic bacteria specifically. Yeah. Okay, so moving on to tetracycline. So the specific drug you need to know for tetracycline is doxycycline. So it binds to 30S subunit. And the main thing you need to know for doxycycline is that it's used for malaria, and it's contraindicated for children under 8 and women who are more than 16 weeks pregnant. And there's quite a lot of adverse effects of this drug. That's why it's contraindicated for quite a large population. It can cause photosensitivity, can calcium chelation, which is just basically affecting like it basically affects bone growth. So that's why you don't give it for children under 8, and it can also cause hepatotoxicity as well as renal damage. So again, a memory tool is quite silly. So the first part of the word dog, so that's like, what do dogs love? They love bones. So that's the whole part about calcium. And cycling, so it's like cycling. So imagine cycling through a park, right? So like when you cycle, it's basically like you can get off the bike, so it's not irreversible, it's reversible binding. And so when you're cycling through the park, usually it's very sunny, so that's photosensitivity, and there's lots of mosquitoes. That's why it's targeted for malaria. And usually you wouldn't recommend pregnant women or young children to be cycling, right? So that's why it's contraindicated for those few. And this is stretching it a bit. So like, imagine the cartoon version of a bike seat and the cycling helmet. They both look like the kidney and liver respectively. Yeah. So that's hepatotoxicity and renal damage. So this specific drug, I couldn't think of a memory tool for this, but if you can think of something, that'll be great. So
Oh wait, yes. Wait, very quickly for this one, my memory tool was chlorine. I think I got this off another PSB. Chlorine. So chlorine, you've got a swimming pool. The swimming pool is 15 meters long, and the adverse effect is gray baby syndrome because the baby drowned in the pool.
Yeah,
A little
that's
bit
good.
morbid.
Yeah. Yeah, basically you need to just know about the gray baby syndrome for this. Yeah. So moving on to nucleic acid synthesis. So the drugs that specifically act on DNA synthesis is ciprofloxacin and metronidazole. And so again, for this drug, ciprofloxacin, it inhibits DNA gyrase, and it's a broad spectrum. And again, so there's a lot of adverse effects. So the memory tool I use for this is fluoroquinolone and ciprofloxacin. So fluoroquine and cipro, right? So this drug is a queen, and it's a pro at ice skating, so you can do many twirls. So basically, that's targeting your topoisomerase, which is your unraveling DNA enzyme, which unravels your DNA. And then it's dangerous for children and elderly to ice skate. So that's referring to your cartilage damage in children as well as your tendon damage in elderly. Yeah, and she's an ice queen, so usually you don't want to expose her to like sunlight. That's why there's also phototoxicity. Okay, so moving on to nitramidazole. So a specific drug you need to know is metronidazole. So again, why you use this, it's for anaerobic gram-positive and gram-negative bacteria, as well as for other parasitic infections. So the one thing you need to know is that it causes metallic taste in your mouth. So like in exams, they like to test certain keywords. So if you see metallic, usually associated with metronidazole. Okay, memory tool. So metro. So that's like the train, right? It's going so fast you can't breathe. So that's why it's anaerobic. And then you have your train tracks, which is your DNA tracks. So your train going super fast on the train tracks will break the DNA strand. And usually you take your train, and that's to avoid backpacking. And so that's why you take this drug for, usually when you see backpackers diarrhea or giardiasis, you're associated with this drug. Okay, another drug which is specifically used for TB, so rifampicin. Okay, so this targets your your RNA polymerase enzyme. And again, like I was saying, you use it in combination with other drugs for TB. And the main side effect of this drug is that it causes red or orange secretions. So memory tool for this is like rifampicin. So like it's pissing. So orange, it causes orange red secretions. Yeah. And R in rifampicin refers to RNA. Okay, so moving on to folate metabolism. So this is the last drug class, and it's something you don't need to know in much detail compared to the others. So the first one, sulfonamides. You don't actually need to know a specific drug for this at your level, and they won't test it. Mechanism of action, again, you don't need to know this in detail, just need to know it inhibits synthesis of folate. And the key thing you need to know about this drug class is that it's bacteriostatic, and it's highly allergenic. So again, I couldn't come up with a memory tool for this, but if you can think of something, that'll be great. Yeah. So THFR inhibitors, again, a specific drug you need to know for this is trimethoprim. So it basically inhibits the conversion of folates to nucleotides, which are your essential part of your DNA. So you've got different drug specificities for different pathogens. So within DHFR inhibitors, you've got trimethoprim, which is targeted for bacteria only. And then you've got methotrexate, which is used for tumor suppression, so for cancer. So just need to know for bacteria specifically, you use trimethoprim. And yeah, it comes to the end of antibiotics. Hopefully it's not too confusing. There's lots of drugs you need to know. And I know you'll probably have to learn it within 2 weeks. And so you probably have to cram it in with all your other knowledge, but that's why I guess the first time learning it, you just try to learn it with some memory tools. And over time, when you go to semester 2, when you're in year 2, you'll start associating with mechanisms of action, and you won't be using as much memory tools in those cases. And hopefully you can reach that stage sooner. But like for now, just try to think of the most silliest things to associate these drugs with, and they'll help you with your memory work. Yeah.
Yeah, I think like another thing is that like Monash likes to test with a lot of keywords. So I think you mentioned like the metallic test. It's going to be metronidazole. If you have like red urine, it's going to be rifampicin. Like things like that. Yeah.
Yeah, and sometimes Monash loves to give like these paragraphs of information, like this. They may give a case study about this whole patient going to see a doctor, and it'll just be full of unnecessary information. You just look out for keywords, like what Peter was saying. And once you see that keyword, usually you're associated with it, with a specific drug. Yeah.
Yeah, like as you start to do more practice questions, or even like practice exams later down the year, you'll start to see like different keywords, and like it's going to be a buzzword, and you're going to know the answer right away from it. Yeah. Okay, sweet. Thank you, Audrey. Who's presenting next?
I can do it.
Okay, antimicrobial resistance. Here we go. What a roller coaster.
Yeah. Field load. Okay. Oh, spoilers. Hello, is it working?
It's just loading. Yeah.
All right, antimicrobial resistance. All right, so essentially, I just already covered with you guys like the specific details of how we actually attack it. But it's like if we think about microbial resistance, right? Like with everything that's currently going on in the world, like on a battle, there's always two sides. Everyone's gonna like kind of fight back. Likewise, antimicrobial resistance. We have all the drugs, right? They're targeting all these different parts of the bacteria. They're very selective of these characteristics, and likewise in antimicrobial resistance, these bacteria, these viruses, these other organisms all can fight back in their own way and resist against the drugs that we give. So, all right, so antibiotics in therapy. All right, so what is it? Well, basically, just think about the bugs getting really chad or like really strong, and then they've like developed this kind of tough skin against the therapeutic agents we use. That's pretty much it. So this is bad, right? Because some patients like they obviously have weakened immune systems, aren't able to actually can't even use any of these antibiotics because, as Ajay said, some are nephrotoxic or neurotoxic, and so then we become very limited into how we can treat. Antimicrobials are also usually selected in choosing characteristics in their microbes and not ourselves. So let's just focus on the fact that these drugs are usually selective so that they don't damage us, but they only damage the bacteria. Back then in like the 1950s, 1960s, when penicillin first came out, actually it was probably a bit earlier than that, penicillin was really effective, but within 2-3 years it already became not as effective. So likewise now, we'll talk about how bacteria has become more resistant, and vancomycin, amoxicillin, cephalexin, these are all kind of drugs that are becoming more and more less and less effective. So targets resistance. All right, so this entire lecture, just a quick overview, is essentially Meredith just covers the main points. She talks a little bit into detail about specific cases, but she doesn't actually expect you to know too much of the specific cases, but just to have it in the back of your mind. So I'll just mainly go over in the main concepts. So in this case, it's like the main targets and how do bacteria resist. So there's innate, there is alteration of active sites, access to target site like altered uptake, and modification or destruction of antimicrobials. Okay, so transformation, right? Like if we think of a Pokemon, essentially what happens is these bacteria, they can transform. And so what is transformation? Okay, so bacteria transform by taking up external DNA so that they become stronger as such. So dead bacteria gives their resistant gene to a live bacteria to help them become more resistant. So, okay, so how do Pokemons evolve? Well, the good way I think of it is I'm not sure if you guys play Pokemon, but you know how Eevee, you have to give the fire stone, the water stone, or whatever spark to go to Eevee to Sparkion, you got the lightning stone, all of these things. So likewise, the stone that we're talking about here is the DNA kind of material that has been given from other bacteria, whereas the original bacteria that's uptaking that, so Eevee, can later on evolve to become this more impressive, more resistant strand of species almost. All right, so what's conjugation? Essentially, conjugation is DNA transfer between two bacteria via a sex pili. Basically, think of this as a bacteria touching through their special parts and then transferring all the big brain knowledge they acquired to one another so that they can survive for another day. So conjugation, think of between two alive bacteria through a sex pili. So alive, so conjugation is alive, transformation is through dead. And finally, transduction. What is transduction? Well, it's a virus that infects a bacteria. So Meredith doesn't really go on much about this. It's essentially a bacteriophage. So it's like one of those alien things. They just sit on the bacteria. And so the virus infects the bacteria. And the virus can have other mutant genes that allows bacteria to become more resistant. Good. So plasmid. So what is a plasmid? Well, this is the actual thing that kind of helps information to be processed and to be shared between different groups, I guess. So different bacteria groups. So plasmid is essentially a DNA molecule, but it's separate from the actual strands of DNA, the bacterial DNA that's kind of free floating. And it's also double-stranded, and the plasmid is transferred through conjugation. I remember you guys in biotech would have talked about how plasmid is also used for like cloning and things like that. So yeah, so think of plasmid as a way to have like these like CD-ROMs, like CD disks that they can just like spread around. So how I think of it is like essentially they're self-replicating, and it's a never-ending donut factory, right? So it keeps making these sort of donuts, like donut wide because it's round, and it's self-replicating because like literally all the donuts like are the same. And the donuts like are separate from spaghetti, right? Spaghetti being the bacterial DNA that's free floating. And since DNA makes you fat and fat protects you from the cold, it's like antimicrobial. So it becomes resistant to the microbial agents that we give. And what are transposons? So essentially transposons is like the way that we can communicate between cells in the bacteria. So it's like from plasmid to plasmid, like plasmid to the DNA spaghetti, like the bacteria DNA, or from chromosome to chromosome. So it's a way for bacteria transposons to kind of jump between different sections of the DNA. And just think of it as kind of a shuttle bus between genetic material. So, all right, so what is the reality of bacterial resistance? Well, they grow really fast because firstly, they can replicate pretty much in 10 to 40 minutes and within hours. So they're getting really busy. Natural selection wise, like if you think about it, if a certain bacteria can survive a certain antibiotic, then there might be some certain genetic variation that cause them to survive. And then they will transfer that genetic information once they die to other bacteria through transformation, or even just through sex pili in conjugation. Practices that can influence a lot of this includes like medicine, agriculture, like veterinary practices when we're using this on antibiotics on a mass scale to protect agriculture, but it's increasingly impacting the bacterial resistance. Misuse, so like you guys will be talking about MindMe as the antimicrobial stewardship. So if you don't actually fully finish your course, like it could cause more resistant strands of bacteria. All right, so boss fight, right? Like everyone loves a boss fight. So methicillin-resistant Staph aureus, or MRSA, is through transformation. And through transformation, we now have BRSA, which is vancomycin-resistant, which is really, really scary, since vancomycin and like carbapenem and stuff is like some of our strongest antimicrobials. I think it's important just to know that you should just know that what MRSA stands for, and it is Staph aureus that has been transformed. You don't need to know too much in specific about it. Meredith does talk about sepsis though, but it's not heavily examined in first year. Vancomycin-resistant enterococci, it's actually a part of the normal flora, but it can become resistant to the vancomycin that we use. And all of these are hospital acquired, so we definitely want to get patients out of the hospital as soon as possible so that they can be, the exposure to these possible bacteria will be decreased. Cool. That I just kind of blitzed through that, but this lecture wasn't there wasn't that much. There was just like I just synthesized the main ideas. Meredith goes into some specific details she doesn't really examine too hard on them. So just understand what's in these slides and like try and break it down for yourself to understand basic concepts, and you'll do fine. Yeah.
Okay, sweet. Let's go through like we have fungal infections, then we have antivirals, and we have some psych left. Yeah,
I think ask them what they want to go through.
Yeah. Which one do you guys want to go through? So fungals, antivirals, or psych? Love psych. Fungal infections? Yeah. If no one else says anything, then yeah, cool.
Okay, I'll share screen. How do I share? Wait, I think someone needs to stop sharing before I share. Who's sharing screen right now?
That's David.
Right.
Yeah.
Okay, thank you. Okay, so let's go through fungal infections. I think I forgot who actually gave the lecture. Was it Meredith, guys? Was it someone else?
Yeah, I think it was Meredith, but I don't really remember either.
Yeah, anyway, so fungal infections, I would say, isn't as high yield. So if you're studying bacteria, viruses, and fungus, and you don't have a lot of time, I definitely skip on the fungus because I just feel like over the years it hasn't really been tested as much compared to bacteria and viruses, mainly because fungal infections are a lot rarer compared to bacteria and viruses, especially the more severe fungal infections aren't as common compared to, let's say, severe bacterial infections. So anyways, first let's go through some features of fungi. So fungi are eukaryotes that are obligate aerobes, so they can produce sexually or asexually. And a few unique things that they have in terms of their structure includes something called chitin in their cell wall. Also these structures or these polysaccharides called beta-glucans. And within their plasma membrane, so beneath their cell wall in their plasma membrane, they've got something called ergosterol. So ergosterol is kind of the same as cholesterol in like human cell membranes. It does the same thing, except they have ergosterol, we have cholesterol. And so these are a few things that are essentially unique to the structure of a fungi. And later on, when you go through antifungals, you'll realize that some antifungal drugs target these unique structures, because what we want to do is we want to kill off the fungi, but not kill off our own cells. So that's why we tend to target things that are unique to fungi. You have three main types of fungi that you'll learn, and these include yeasts, molds, as well as dimorphic fungi. So let's go through the types first. So this is a bit of a summary table. So we have yeasts. I think a lot of people have heard of yeasts before. So these guys essentially reproduce through a process called budding, where essentially there's a developing bud, it grows bigger, and then at some point in time, your new daughter cell will just bud off and it'll split into two. And yeasts are unicellular. And what essentially these little yeast cells can do is that they can link up with one another to form something called a pseudomycelium. So if you see the word pseudomycelium, that just means a long chain of yeast cells, one after the other. And examples of yeast include Candida, Cryptococcus, and Pneumocystis. Moving on to molds, once again, I think a lot of people have heard of the word mold. So molds are different from yeasts. They essentially grow or reproduce by these extending hyphae. So You have a septum, and then a new hyphae essentially branches out, almost like a tree branch. And as these grow, you form something called a mycelium, and this is essentially just a huge, large mass of hyphae all entangled together with one another. And molds are multicellular, so that's the main differences between yeasts and molds. Molds also can produce spores, and which can then be breathed in. And that's one of the ways that we can essentially get infected with molds, is when we breathe the spores in. A very important example of molds to note is Aspergillus, which can also cause allergies, and we'll go through that in just a second. Now, the last main type to note is dimorphic. So this just means, so di means two, that just means that they can essentially morph between a yeast and a mold form. And typically they'll change their form depending on the temperature. Typically, they'll be mold at around 25. When the temperature increases to 37, they can become a yeast. So examples include Penicillium, Candida. The only one to note is Sporothrix is dimorphic. However, their temperature is actually the other way around. So Sporothrix is a yeast at 25 and a mold at 37. Okay, so fungal diseases. So how do fungi actually cause disease? They can cause disease through a few ways. So one way is through allergies, and this is our Aspergillus, which can cause something called bronchopulmonary aspergillosis. That just means we've breathed in the Aspergillus into our lungs, and we have an allergic reaction that goes on in there. Some fungi can also produce toxins, and that's one reason why we don't want to eat food when it's expired, because if there is something like mold that grows on there that produces toxins, we don't want to be ingesting that. And the next one, which I think is common sense, and that is infection. So these can either be cutaneous, subcutaneous, or invasive. And your lecture will mainly focus on the different types of fungal infections. So that's what we're going to go through as well. So fungal infections, another word for it is mycoses. So let's go through the different types. We'll start off with the superficial one, or the cutaneous. And superficial just means that the fungi will stay on the surface of your skin and they won't go any deeper. The reason why they stay on the surface, such as your skin, your hair, or your nails, is because a lot of these fungi actually require keratin to live. And your skin, your hair, and your nails is full of keratin. And that's why they like to stay there essentially. Of course, if they cause a really dry skin and your skin ends up cracking, this can lead to secondary infections because you've essentially just broken that first innate natural barrier. And a lot of these superficial fungi will prefer warm humidity and especially sweaty creases, so in places like your armpits, for example. And a common type of fungi that causes superficial infections is tinea. So tinea rubrum causes what's commonly known as athlete's foot, but of course other types of tinea can also infect your head, so more specifically your scalp, as well as other parts of your body. Diagnosis, I wouldn't really say diagnosis is such high yield, but if you would like to know, it's there, and typically we will treat it with azole antibiotics that you guys have just gone through. So now we've gone through on this, what happens on the surface of your skin. So if you go one layer down, we get into our subcutaneous layer. And so subcutaneous infections will mostly occur when there's an inoculation. For example, something that pricks open your skin. And so key buzzwords that Monash likes to use is one, inoculation. So that might signify a subcutaneous fungal infection. And especially if you hear that someone has been gardening and they've essentially got a wound from a rose thorn, that could indicate a type of fungal infection called Sporothrix schenckii. So that's like buzzwords for Sporothrix schenckii. So this is a type of fungal infection that's sustained after inoculation, as we said, very commonly from a rose thorn. It can stay local, but once again, because we've essentially broken into our skin, it can spread to other areas through lymph, and that's why we get essentially several red blotches all throughout the skin, as you can see in the picture there. Once again, it's treated with an azole antibiotic, or you can give saturated potassium iodide. That is not high yield at all. Okay, so moving on to, I guess, the really bad or severe fungal infections, which is your systematic or deep infections. And that's because it involves more of your internal organs, and they typically are more invasive. And so the main ways that we essentially get a more systematic or deep fungal infection is we either breathe in the fungi so that it enters our internal organs or our lungs, or a nosocomial infection. So when you get at the hospital after, let's say, through a catheter that maybe wasn't the cleanest. So Candida is one type of fungi that can cause a more invasive infection. So Candida is actually part of your normal flora, but it's a very opportunistic pathogen. And I think a key or a buzzword, at least for first year for a Candida infection, is when the patient essentially presents with symptoms after they've had an antibiotic treatment for some previous bacterial infection. Because when you give someone antibiotics, you're killing off all their natural flora or their normal flora, and then Candida goes, yay, all my competition is gone, I can start infecting this person. And there's a high mortality rate with invasive Candida because it's quite resistant to drugs. And those are just a few types there. So the main things that Candida causes is oral thrush as well as vaginal yeast infection. And this is especially common in women who have undergone previous antibiotic treatment. That's probably one of the most high yield keynotes to remember for Candida. But just to note that I've listed Candida here as one that causes an invasive infection, but Candida infections can also be cutaneous on the skin. Another fungi that can cause an invasive infection is Cryptococcus. So it's the small budding yeast, as we mentioned before, and because it's invasive, it's got high mortality rates, so above 50%. And these are essentially a few different types of Cryptococcus. Essentially, just to I guess the take-home message here is it commonly causes meningitis. So the two types here we have neoformans, and this typically comes from bird feces. That's a common pathway of how you get it, and it causes meningitis and sometimes lung infections in immunocompromised people. Whereas Gatti or Gattai, this causes meningitis and it can actually infect healthy people. And once again, the diagnosis you can do through an Indian ink stain and a culture. And for those who are not quite sure what meningitis is, that's essentially the inflammation of something we call the meninges, which is essentially the outer layer of our brain and spinal cord. So it's a layer that surrounds our brain and spinal cord. Okay, continuing on with invasive infections, another fungi that can cause that is Pneumocystis. And it commonly causes something called Pneumocystis pneumonia. So it causes a type of fungal pneumonia, and it's actually quite rare in healthy people. So you don't normally see a pneumonia caused by fungi in healthy people. However, if you are immunocompromised, for example, if you have HIV or AIDS, then it's a lot more common for you to get Pneumocystis pneumonia. And your symptoms for this would be quite obvious. You get a lot of pneumonia symptoms such as shortness of breath, dry cough, fever, weight loss, etc. And then we also have Aspergillus. I think Aspergillus is just, the word is thrown around a lot because it can cause a lot of things, and it's also one of the more common fungi that can cause a fungal infection. So a lot of people tend to think that it's part of our normal flora, but it's actually not. So that's important to note. And so they typically infect our lungs and sometimes our brain, but mainly remember that they can infect our lungs. So Aspergillus, as we said before, it can cause disease through an allergy, but it can also cause disease through, as we see now, an infection. And typically it's an invasive infection because we breathe in the Aspergillus spores, and that's how it commonly infects our lungs. And because it infects our lungs, for diagnosis, we can do microscopy or we can culture it from our sputum. Okay. Okay, and that was a quick rundown of fungi and fungal infections. So, yeah.
Alrighty. Yes. So
Wait, hang on.
is it not me?
No, I'm just thinking.
For how long?
I'm just thinking, like, how long will... I'm just thinking, like, how many slides do we have for antivirals and antifungals?
It will be very, very quick because there's really nothing to go through.
Oh, okay.
Yeah.
Okay, cool. Sweet.
Okay, so, okay, we'll go through this. Antivirals and antifungals, there is actually not that much to it. So virus review, so I think we went through, hopefully we went through this last week. So you've got like different phases of like viral entry and pathogenesis and things like that. And the different viral drugs that we use just target different phases of that process. Importantly, we have neuraminidase, which is the thing that helps the virus particles exit the cell. So next one. So the important one is oseltamivir. The brand name is Tamiflu. I like some brand names. Some brand names are actually very entertaining. But yes, this one's called Tamiflu. This one inhibits neuraminidase. So it inhibits this enzyme right here, which allows the virus to exit the cell. So basically the virus just remains trapped within the cell and it can't escape and infect other cells. And this is mostly just used for the treatment of influenza A and B and for the prophylaxis of influenza. So this was actually it was interesting because this was used a lot in the start at the start of the year when COVID was a thing. I mean, it still is a thing, but it kind of went out of stock and people were kind of panic buying it for all the different aged care homes. It's just a fun fact. Next one is nucleoside analogs. So if we go back, we are now talking about, it's not on here. Is it? Oh wait, no, it is here. Nucleoside analogs. So this is these are drugs that interfere with the process of nucleic acid synthesis. So the main one you need to know is aciclovir, and this is just a guanine analog. You don't really need to know that, but you need to know what's been bolded, which is that it inhibits viral DNA synthesis. So this is used for varicella zoster, so shingles, as well as herpes simplex. And the adverse effects are not that significant. So basically, when we're talking about adverse effects, you have to just remember all the really significant ones. So for like cefalexin, you have to know that it has neurotoxicity, and I don't know, like aminoglycosides can cause like nephrotoxicity and stuff like that. But like in general, basically all drugs cause GI issues and headache. So if a tutor comes up here and they say, oh, what adverse effects does this drug cause? You can basically always say GI issues and headache, and you'll be right 99.9% of the time. Okay, so those are the two first main drugs. So this one's for influenza. This one's for like varicella, zoster, and herpes. The other main group of antiviral drugs you have to know are antivirals used for HIV. So the whole thing is retroviral therapy. So the aim is obviously to reduce transmission, restore. You can read through it. I know you can read. And general principles of treatment are that you generally want to have combination therapies. So you want to use different drugs in multiple classes. So this will prevent resistance as much as possible. And you also want to use fixed dose combination medications as much as possible. So these are like when you combine different drugs into one single pill. And that's important because it ensures that it just makes it easier to take. So if you're taking one pill instead of like 5 throughout the day, people are obviously more likely to actually comply with their treatment. And non-compliance is bad because HIV is bad and transmission is bad. Okay, moving on. HIV protease inhibitor. This is like one of the main ones. So the whole mechanism of action is kind of confusing for this one, but basically it's just, you can just remember what's bolded. It's a viral maturation inhibitor as well as a pharmacoenhancer. So what it means by pharmacoenhancer is that, we'll go into the notes. It says the potent inhibitor of CYP450 enzymes. So CYP450, hopefully you all know, is like a system of, enzymes within your liver which break down drugs. And basically this drug kind of inhibits the breakdown of certain drugs. So what it means is that if you use ritonavir, or however you pronounce it, together with other antiretrovirals, this drug will stop the breakdown of the other drugs because it's inhibiting the CYP450 enzymes. So it'll kind of boost the effects of these other protease inhibitors. And that's kind of what this whole thing means. I was very confused about this last year. And other than that, it's just got some general GI effects and some taste perversion and all that kind of stuff. So these are the adverse effects, not very high yield at all. Okay, last one I think for, yes, antiretroviral drugs is this drug called Truvada. So it's a combination drug like the ones we were discussing before. So it's a combination of this and this. I'm going to try to pronounce it. And what they do is that they block reverse transcriptase, which is something you need for, well, something the HIV virus or the HIV virus needs to like replicate. So that's that. And interesting thing about it is that it can be used for HIV prophylaxis. So hopefully you've all heard of PrEP before. So people who engage in things might need to take it prophylactically, and that's good. Another important thing though is that taking this alone, if you're HIV positive, is not enough. So it's enough to kind of prevent HIV, but it's not enough once you have it. So you'd probably take Truvada plus some other antiretroviral drugs. And there's some adverse effects and precautions, not very high yield at all. Okay, next one, antifungal drugs. So this is kind of going back to what Jiting kind of talked about, but the main thing with antifungals is that we're targeting that cell wall. Why are we targeting the cell wall? Is because it's different to the human cell wall, because it's got ergosterol or something in the cell wall. So that one is, only in fungals, so it won't kill our own cells. And that's a good thing. So main drugs, our main class of antifungal drugs are azole antifungals. So main one you need to know is fluconazole. So I don't know if any of you have ever gotten like yeast infection or what's it called, thrush, but you can actually take a tablet of fluconazole and they'll kind of deal with it. Basically, you can remember that part, or you can just remember the fact that it messes with cell membrane synthesis. And if it messes with cell membrane synthesis, it'll just kill the fungus and stuff like that. It can be used topically and orally. So there's a fluconazole cream, and you can also take a tablet, and it's used for fungal infections as well as meningitis, as Jiting just mentioned. Adverse effects, not very many. You can have some GI effects, and there are some CYP450 interactions. Okay, I think this is the last one. Oh, no, it's not. The two more. This is the second last one, choline antifungals. So you can have amphotericin B. So this is quite similar to the previous one. So it binds to ergosterol in the fungal cell membrane, or maybe also in cell wall. And it alters cell membrane permeability. That leads to cell death. And this is generally used for more severe systemic. So not like if you have a fungal infection, let's just say down there, you would just use fluconazole, but amphotericin is generally used if it actually spreads to your blood or it's like becomes more systemic and it's more serious. This is where you pull in amphotericin B. And there are some issues with it. So it can cause nephrotoxicity, so it can mess with your kidneys, and there are some infusion reactions because it's usually done IV. And more general fever, chills, nausea, GI effects. Okay, last one is caspofungin. So once again, it affects your cell wall. You can kind of see the theme here, and it's used for aspergillosis. This is not a very high yield one, but it's on your e-pharmacology list, so you should know it anyway, and it can cause GI upsets, fever, and flushing. And that is basically it. I just finished making the quiz for next week, so you guys should all study up on your drugs. It's going to be a fun one. Okay, very nice.
Cool. Sweet. I think that just leaves me left. So it's like it's a lot lighter for me. I'm just giving psych stuff. It's like, I don't know how interested you guys are in psych, but sure is my favorite. Love it. I'll just bring up the slides, but does anyone have any questions in the meantime about anything? Otherwise not. Otherwise we'll try rip through this because yeah, it's fairly not too bad. I'll just share screen. Okay, sweet. So being ill, it's like in one of the series they give basically. And it's talking about like what we do when we get sick. So whenever you get sick, like it's the middle of winter or something and you wake up with a sore throat, right? You're gonna have like a decision making to think like, oh, should I go to the doctors to get this treated? Or can I just sleep for the whole day and will I be okay, right? And this will be like a decision-making process. And you'll think about a few things. So you think about like the problem. So what's the problem here? Like, do I just have a sore throat, or is that sore throat stopping me from going to school or something like that? What type of intentional action can I take to treat it? So like, am I able to, I don't know, drink tea and sleep? Or am I literally unable to do anything? And then the other part of it is that it's often interpersonal. So thinking about like, oh, should I tell someone about this? Should I go see the doctor? That kind of thing. So we have all these things to consider when we're trying to figure out whether or not we should access help. Our likelihood of accessing that help though is increased if it's like perceived as serious, if it's painful, if it's really disrupting our life and things like that. A short bit, illness delay is just the time between like you get a sore throat and then you go see the doctor. So that time period, once again, can be shortened or lengthened depending on how much pain there is, how severe you think it is, whether you have like family history of it, like how worried essentially you are. The more worried you are, the shorter delay you'll have. I'm sure everyone's experienced this. Illnesses can affect you a lot. So if you're really sick, you're unable to do anything. You don't know if you can go out for dinner tomorrow night. Don't know if you can go to uni. Am I able to go get a coffee? Things like that. But it's also going to have a few more positive things. So it's also going to help you to strive for recovery. So for example, whenever I get sick, it's probably because my sleep schedule is really messed up. So when I get sick, I try to fix my sleep schedule so that I can get better sooner, and then I can go back to my bad sleep schedule afterwards. So it kind of helps you strive for recovery, helps you become a better person. Also helps you take care of your health better in the future, hopefully. I forgot who gives the lecture for this, but he brings up this theory, and it's like a sick role theory. So what are people like when they're sick? And once again, this is pretty easy to remember. You kind of just think about to the last time you were sick and what you were able to do. So for most people, when they're sick, you don't have to go to uni, or you shouldn't really go to uni, but you don't need to go to work. You probably don't need to do the cleaning as much, things like that. People often don't blame you for getting sick. They're probably like, oh, someone else was sick and they gave it to you, rather than you went out and caught the cold yourself. The person who's sick wants to get better, and they're going to cooperate with people trying to give them help. Like they're not going to resist someone telling them to go take a nap. Like if someone tells me to go take a nap because I'm sick, I'm just going to listen to them because their help is really, really good. And then as the sick person, we're going to view our sickness in a certain way. And depending on how we view our sickness, this is going to affect on whether or not we get help for it. So if I wake up with a sore throat, I'm probably not going to think much of it because it's simply just a sore throat. It's happened so many times before, I don't really care. But if I wake up and I can't move my right leg, that's going to freak me out a lot. So my perception of my illness is going to cause me to go seek out help a lot quicker. And how we perceive different illnesses can be affected by a bunch of things, namely our culture, our family, our past history with that condition, whether or not we've got it before, and how it turned out then. So really, a lot of it depends on our past. And then if we're sick, we have to consider our quality of life. So how would we rate our life in relation to what's going on around us? So am I happy with it? How much am I enjoying life? There's a definition from the WHO, don't really need to remember it though. So quality of life is actually really important because although it's not related to pain or anything super quantifiable, it's really important. It's really pertinent for the person's mental health, because even if someone has no pain, they're feeling well, but they have a really terrible quality of life, you've kind of missed the whole point of your healthcare. It's meant to be patient-centered. It's meant to help the person live their life better. And if their quality of life has gone down the drain, there's no, like, you kind of failed, honestly. So you really should be like looking holistically. Although quality of life can be affected by other things. So depending on your illness and your treatment, so what side effects you're getting, your context and your coping and your social support. If you have more coping strategies, more social support, you're going to have a better quality of life. And luckily for social support, we consider carers in Australia. So those who take care of us when we're sick or when we're feeling ill. So it's really quite beneficial if you have a carer or if there's a carer caring for someone who's sick, because it can improve the adherence to treatment. It feels like someone is going through the illness with them and they're not alone. So this helps them have better adjustment, better coping. Generally, like with social support, I think your immune system also gets better. You can probably ask, I don't know, like Craig Hassett about that. And generally, it just improves things. But if you think about it from the other side of it, you have to be mindful of the effect that that caring can have on the carer. Because if your job every day is to take care of someone, bring them to the shops, shower them, bathe them, cook for them, clean for them, go to doctor appointments with them, it's going to be really, really draining on the carer. So you have to think about the burden that it places on the carer and how you can manage that. Because if you're unable to have a long-term carer, then you can't really form that social support with that person. So you might consider like fostering things like that, or taking personal time off on the carer's side. And then lastly, prevention and change kind of focuses around like the Prochaska-Di Clementi changing circle thing. So behavior change is really important for patients because you want to get rid of risk factors and you want to improve protective factors. There's also another approach called the health action process approach. And it's kind of made of two stages, the motivational stage and the volitional stage. So the motivation is what the name suggests. So it looks at our own motivation and how much we want to change. It's like looking at our intention to change. And we consider a few things. We consider what's good about changing and what's bad about staying the same. And we also evaluate how well we think we'll be able to change. So do I think that I have the ability to change? Maybe. And after we've passed the motivational stage and after we've determined like, oh yeah, I want to change, we have to look at the volitional stage. And this volitional stage is kind of where the action and the change occurs. So we make a plan, we think about how confident we are in our own ability to deal with the difficulties that it might bring along. And then we actually do the change. So we engage in the behavior. But with all changes in behavior, we're probably going to fail at some point. So we also have to think about our own ability to deal with those failures. So as an overall picture, stage 1 is to do with the intention, stage 2 is to do with behavior. And that's why it helps to bridge the intention and behavior gap. Because intention to do something and the actual doing of something is quite different, and this helps to bridge that gap. This is what it looks like in diagrammatic form. This diagram really didn't make sense to me. Yeah, but you can kind of see like these purple ones here on the left contribute to the intention. And then after you've done the intention, you kind of move on to planning for the action and then you actually do the action. And you have a bunch of self-efficacy, so like evaluating your ability to do something in the planning stage and the action stage. Yeah, I wouldn't worry too much about the diagram. And then last one is the Prochaska and DiClementi cycle change. So you have your 5 steps, your PC-PAM. I think we've gone through it, but pre-contemplation is like, you're living in denial, you don't need to change at all. Contemplation is, oh yeah, I might change. Preparation is like, yep, I'm going to start to change. I'm going to, I don't know, get rid of my loyalty card to whatever restaurant I'm going to. Get rid of my loyalty card to the cafe. 4 is the action actually occurs, so you may not go out to eat as much. Then 5 is maintenance. So keeping that change and prolonging it. But obviously you're also going to get relapse. So you might fall back between maintenance of your new behavior back to your old behavior, and that's okay. It's going to be a constant cycle of trying to fight back relapse. Some criticisms of the model. It's like these 5 stages, although they're portrayed to be separate stages, a lot of these stages can blend together. The model also doesn't give like a specific timeline. So pre-contemplation could be like 50 years before contemplation occurs. And then even between contemplation and preparation, that could be like 5 years, or it could be 5 minutes. Different people move at different speeds. And this model doesn't really give individual variations. So it doesn't really consider a lot of factors like your social factors, talking to other people, things like that. So although it's a good framework, it doesn't fully represent everything. Yep, so attitudes, they're like our feelings about certain things. And often our attitudes will lead to a behavior, but that's not always the case. So for example, if I have an attitude that I really like coffee, that attitude of really liking coffee will often lead to the behavior of going out to buy a coffee or going out to make your own coffee. But if there's a discrepancy between that, like, oh, I really, really like coffee, but I refuse to drink it, that simply does not make sense. So that's where we have cognitive dissonance. And it's like your 2 people sitting on your shoulders and they're like, you really like coffee, but don't get the coffee. So there's a bit of fighting between there. So as I said, attitudes often predict our behavior, but they don't always. That can be due to a variety of factors. Often peer pressure and your social influences will play a large role in that. So you want to fit in, and therefore, even if you have a different attitude, you may not show that attitude through your behavior because you want to appear likable and things like that. However, if your attitude is really, really strong and you're really firm in your attitude, yeah, you probably won't let that happen, and it'll probably predict your behavior. Okay, there's more than I thought. But persuasion is like when someone tries to change your attitude. And I guess the most important part of this is the 3 components of persuasion. So the source, the message, and the audience. So the source refers to who is trying to change your behavior, who is trying to change your mind. Do you like them or do you not like them? The message is like what they tell you and how they tell it to you. Is it, yeah, are they giving you facts or are they just like screaming at your face? And then the audience is the recipient. So the recipient, you have to consider the recipient because the recipient could be closed off to the idea or they could be open to the idea. And depending on that, their likelihood of being persuaded to change their attitude or their opinion will differ. So the best persuasive messages tend to arouse some sort of fear. They emphasize like the ability of the recipient to change. So it's giving them confidence and hope that they are actually able to change. And that's what occurs during motivational interviewing. And then lastly, this is a model we think about when we think about persuasion. And we have 2 routes through which you can persuade people. So a central route is where you have an audience that is really engaged. So they're listening, they're actively listening and trying to think about the argument and why they might change. So these people actively want to change and they're listening to you. But the peripheral route is like people who don't really care, but they're simply listening to you because they feel like it's kind and it's polite. So they're not really evaluating your message. It's just going in one ear and coming out the next ear. So it doesn't really result in any change. So central route is better, but it's a bit more difficult. Peripheral route is worse for change, but tend to be easier. And a lot of people go through the peripheral route. Yeah, and that is our psych, and that's the end of the week 11 content, which was quite a lot, honestly. Yeah, that was a lot. So, yeah, cool. Does anyone have any questions to finish up, or oh, you're good? All good. Cool. Yeah, that was intense. Jeez. Anyway, good luck for your last OCEs this afternoon, I think, like giving an explanation. Yeah, good luck for those. Hope you guys are doing all right with those. Otherwise next week is pretty light, if I'm not mistaken.
Next week is like nothing.
Yeah, next week is HEPN, like HKS. It's like all your acronyms. So.
Oh, I need to do HKS. Oh shit.
Yeah.
It's my session.
Yeah. Yeah, so once you guys make it through this week, like you've made it, you've basically finished semester 1. So that's all good. Otherwise, yeah, we'll catch you guys like next week sometime. Try to figure out a time.
End of sem revising. Do we want to run an end of sem revision? We probably could.
We probably could. We'll talk about it. Yeah, you do. Yeah, we'll see. We'll see about it. We'll talk about it. Yeah. All right, cool. Catch you guys next week.
Thanks for coming, everyone.
Bye-bye.